Hello, AP Bio. In this video, I'm going to walk you through how to do some of the genetics practice problems. I apologize. I've been having some technical difficulties, so I'm just going to show you some pictures of my notes and I'm going to talk you through how to do them. It's not ideal, but that's what we have to work with today. Okay. So I think all of you are able to solve the one trait cross. There's a couple things that I just want to make sure just to patch through for some of the blocks that we didn't get to it yet. You should be able to, I needed to define some of these terms in here. For example, a carrier is just a heterozygous individual who carries in a recessive allele, but they don't express it. So it has to be a trait that follows a complete dominant pattern of inheritance and a Mendelian inheritance, and that trait has to be able to be masked. So it has to be a recessive trait. And a carrier is just an individual that carries that recessive trait, whatever it is, and then can also mask it. So they don't express the trait. So they themselves are not affected, but they carry that recessive allele. The term hybrid refers to a cross of two varieties. In the case of genetics, if we're looking at genotypes, it just means a heterozygous individual. So a hybrid individual is synonymous with a heterozygous individual. Hybrid doesn't always mean that, but in the case of genotype, that's what it means. Pure, on the other hand, is basically the opposite of hybrid, which just means a homozygous variety. So it's the same allele at both loci. So capital A would be a pure. Purebred, thoroughbred, those all mean the same thing. And the P generation in a specific language to a Mendel's cross would always be pure. But the problem will tell you that, like a pure P generation or whatever. Anyway, okay, so I think you should be armed with completing questions all the way through number 12. We're going to talk about the rhesus factor in class, but you should be able to solve at least this problem right here because it's just complete dominance. Keeping in mind, I'm wanting you really to think about your symbols that you use. So your symbol that you use should drive whatever the dominant allele is. So do that. Okay, so now let's move on to test cross. Let me walk you through how to do a test cross. Remember from the video that you watched. So the test cross is all that it is a cross between a test individual and homozygous recessive. So in this case, a farmer's told that there's a black bull is thoroughbred. So you're going to try to test to see if he's thoroughbred or if he's being duped and maybe he's heterozygous bull. Knowing that black cattle color is dominant over red, he decides to determine the purity of the strain by matching the bull with several red cows. If the bull is pure, what will happen? Provide evidence. Okay, so it's just a genetics problem. We got to start with the key. We'll use capital B for black and lowercase b for red, and then we're going to set up our test cross. Remember that our test cross, we're going to have the test individual. It's a bull, so we'll put it first. Male goes first by convention, and then the female. Here's the cow. And the cow needs to be red because it's a homozygous recessive individual in a test cross. So here we're testing for this allele. We know that the bull has to have one big B because he's black, and we just don't know what this is. So we've got our cross right here, and now we're going to set up our Punnett square. Our Punnett square will put the male across the top and the female on the left, and then we're just going to fill all of our possible zygotes. Here we'll test to see if the bull is thoroughbred. It wouldn't be wrong to test to see if the bull was heterozygous, except that would be, this is sort of the null hypothesis, saying, okay, it must be, but you could do the alternative hypothesis too. Either way, you would just have to reason why you were testing one or the other. But if you're testing for thoroughbred, then your expectation should be that you would get 100% black offspring if the bull was thoroughbred. There's no way you could get anything else because all he would be giving away in his sperm were big B, big B. And in that way, you would know that it was a thoroughbred black bull. 100% black offspring would be your evidence that he was thoroughbred. Keeping in mind that you'd have to have a large sample size. If the sample size was small, then you might, by chance, just get 100%. Okay, now you can try number 14 on your own. And after you try number 14 on your own, then you can come back to this video, or you can listen to the whole thing right now. It's your choice. To get ready for calculating probability and to complete section 3, there's a few points that you need to think about, or rules about calculating probability, considering that really all that a Punnett square allows us to do is calculate probability with the model. So let's just think about what those numbers mean. Well, the reason that Punnett squares work, and the reason that Punnett's math works, is that all organisms and all somatic cells are diploid, but gametes are haploid. Each allele, if we think about it, it's a haploid allele. It has a 50-50 chance of being carried by a gamete. So that means that 50% of the time it's going to go one way and 50% of the time it's going to go the other way. It's just that it's either maternal or paternal, thinking about that where every single allele you have 2 because you are diploid. Now, Punnett squares, what they actually are, is a model of predictive probabilities, but of independent assortment. Because consider that it's not really 50-50 because those alleles are living together on chromosomes, and chromosomes travel as like big giant molecules, right? So keeping that in mind, sometimes things aren't a 50-50 chance if they're traveling together. So we're going to get to gene linkage later in the unit. But you should, for right now, just be aware that Punnett squares are modeling only independent assortment. Now, how do you use probability, and what are the laws of probability? We're only going to learn 2 rules of probability, so don't get yourself all worked up about not knowing probability. You're going to just calculate each locus separately. So we're either going to use the multiplication rule or the addition rule. The multiplication rule is anytime you see the word and, like if 2 things might occur or will occur together, then you're always going to multiply those probabilities together. On the other hand, if you see one thing happening or the other thing happening, you're going to add them together. So let's look at an example of that. A skin or abnormality in humans, keratosis, is a dominant trait. A man with keratosis, whose father did not present with the abnormality, marries a woman with keratosis, whose mother also did not present with the abnormality. If they have 3 children, what are the chances that all 3 children will show the condition? Okay, so now they have 3 children, all 3 children. So it's the first child and the second child and the third child. So now we're going to use the multiplication rule. We're going to solve these just like we solve all other genetics practice problems. We're always going to start with making a key. It tells us that keratosis is dominant, so we'll use capital K for keratosis and then lowercase k for normal. We're going to set up our cross. It says a man with keratosis, so we know he has to have a capital K, and his father didn't have the abnormality, which means that his father was little k, which means that he has to have a little k. So he's big K, little k. And the woman has keratosis too, so we know she is capital K. By the way, remember male first and then female. So we know she's capital K because she has keratosis, and her mother didn't have it, so we know her mom was little k, so she must have a little k. So there's 2 hybrid individuals make a baby. Okay, here they're now having they're having their own children. So now this is what their kids look like. Here's the mom, and here's the dad right here. And the question asks, what are the chances that all 3 children will show the condition? Well, again, it's a dominant allele. So this offspring right here, this child will have it, this child will have it, this child will have it, this child won't have it. So when you look at that in a fraction or in probability, 3 out of 4 are going to have keratosis. So the first time they have a baby, the chances of them having a baby with keratosis are 3 out of 4. And the second time, the chances are the same every time because all their germline cells that go through the process of meiosis are all the same. It's always the same for those germline cells. So it's 3/4 for the second time. It's 3/4 for the third time. And you just and multiplies those numbers together. You get 27/64. Okay, that's a super weird number. No one would ever say, well, 27 out of 64. Like, what? And so typically, if you get a weird number like that, you would want to convert that into a fraction, and you'd say 42% of the time, if you have 3 kids, the chances that all 3 of your kids will have keratosis is 42%, as opposed to 27/64 of the time. Like, nobody talks like that. Okay, anyway, let's do it again. Let's now look at adding 2 traits together. If they have another child, what are the chances of it being a girl and, there's the word and, an individual without keratosis? Well, first we have to do the probability of having a girl. The only time we break tradition with Mendel's law, Mendel's symbol system is when we're doing the sex chromosomes and you just use the actual sex chromosome for the symbol for the allele. So female goes on the left, biological female, that's XX, biological male goes on the top, XY by convention, and here's our Punnett square. Female's on the left here, biological male's on the right. So the chances of it being a girl, 1 out of 2, you can see that it's 2 out of 4, which reduces to 1 out of 2. And an individual without keratosis. Well, if we look at our Punnett square of the individuals without keratosis, that's a quarter. So a half times a quarter is an eighth, 12.5%. So that is how you would solve probability problems. Okay, so you can go ahead and do 15 through 19 on your own. And then you can come back to the video, or you can just keep listening to me blather on about another example to try. Another example, another type, non-Mendelian inheritance, another type of genetics problem you might see are when we look at degrees of dominance. So remember, with degrees of dominance, it's non-Mendelian. So what we're looking for is more than 2 phenotypes. And for both incomplete dominance and codominance, we're looking for 3 phenotypes. And in these practice problems, that's what you'll see. It's a red flag that, oh, this must not be Mendelian. So I don't have complete dominance, I have another type. So in question 20, the question says this: crossing a plant bearing white flowers with a plant bearing red flowers produces pink offspring. This observation is evidence of, well, you see white and you see red. Remember that incomplete dominance is a blend of the two, making pink. And so that should be, oh, that's just a definition thing. This observation is evidence of incomplete dominance. But right away, it should be a red flag that it's not Mendelian inheritance. It's not complete dominance because there's 3 phenotypes. And incomplete dominance, remember that neither phenotype is expressed in the third phenotype. It is really just a blend of the two. Number 21, let's look at a specific example of how to solve an incomplete dominance practice problem. The question says this: when round squash, 1 phenotype, are crossed with long squashed, 2nd phenotype, all offspring are oval in shape, 3rd phenotype. Bam, it's not complete dominance, and it's really a blend of the two. Oval is kind of round and long blended, right? So I know it's incomplete dominance. How many genotypes are produced when round squashed are crossed with oval squash? Well, first thing we have to do is make our key. So when we do incomplete dominance, they're both, you can think of them as both being recessive alleles. So we're going to use the lowercase superscript. So SR for round and SL for long, both lowercase. And then the only difference that we're going to do for our key when we're looking at degrees of dominance other than complete dominance is that we're going to show the heterozygote and we'll show what the 3rd phenotype is. Again, you want to make sure that you're writing those in alphabetical order just to keep yourself organized. So here's our key. And then it says a round squash individual, and there's no male versus female, so we're just going to put the first one first. A round squash individual, it has to be homozygous for the round allele, and is crossed with a long, an oval squash. An oval squash is always going to be the heterozygote. And then, so we have the SL, SR in alphabetical order, and then we're going to just distribute those into our Punnett square. Next step is to make that Punnett square. And in our Punnett square, we'll do, across the top, we'll have our round squash across the top, and then we'll have our oval squash across on the left. And you'll see here that you're just going to fill in your Punnett square, and then you're going to get 50% of your squash are going to be round. You can see them here, and 50% of your squash are going to be oval. Okay, now you can try 22, 23, 24. In 25, it's asking about codominance. And so 25 and 26 are codominance questions, and you can read this question here. Just make sure that you see when codominance is they're both going to be expressed. Okay, in part 5, I want you to start thinking about multiple alleles. So in multiple alleles, we have a situation where you have more than 2 choices for each locus. Blood type is a classic example of multiple alleles. In the ABO blood typing system, we have the A allele, which is really, it's going to be codominant with the B allele. So we're going to use a capital I. It's immunoglobulin. It's just by convention we use I. And then superscript A for the A allele. We're going to use superscript, capital I, superscript capital B for the B allele. And then for the O allele, it's actually a recessive allele. So there's 3 different choices in this allelic series. Remember that about allelic series. And so we have the A allele, the B allele, and the O allele. It says this: one parent is heterozygous for type A blood.